Yes. So the first question for today is the microarray data was collected from a wild type Arabidopsis plant and the mutant plant lacking lateral roots. What what could be concluded from the data? So microarray is basically used to differentiate or between different organs or different development in stages on the basis of their transcriptome profile. So the options are differential proteins in the mutant plant, differential post-translation modifications in the mutant plant, differentially expressed mRNAs in the mutant plants, and none of the above. Just a second. So the correct answer is option C, that is differentially expressed mRNA levels in the mutant plants. So first we'll discuss what is microarray. So what happens is like it's basically a chip kind of thing, and in this chip various, like this is the chip and various spots are made on this chip, and in each spot there will be the all the genes of the organism or the cell that we are studying. So after we add a RNA or the cDNA, that is reverse transcript from the RNA that we isolated from the sample, that is labeled with either a fluorescent dye or a labeled dye that we use in one color array. So after that, hybridization occurs between the cDNA that we have isolated from an organism or a cell, and thus the DNA that is present in the wells that are present on the array. So the data is then analyzed, and after that we see what are the differentially expressed mRNA are present in the cell or an organism. So here is a one color array. So in there is a test sample and a control sample. So first we isolate the mRNA, and after that we generally we do a reverse transcription, and using that we use labeled probes here. So we used biotin here for labeling, and the cDNA that is formed is biotin labeled. After that we put these labeled cDNA onto these chips and in each well, and that each well constitute the cell from which the RNA is isolated. So after that scanning is done and data is analyzed, and after that we can find out which gene or which mRNA is expressed more in case of the mutant or the wild type. Now what is the second one? This is the two color array. So in this, what is the major difference between these two is, here we'll be using two different fluorescently labeled dyes. So if we are analyzing one sample, so it will be labeled with a different color dye, and the other will be labeled with a different colored dye. So after hybridization we'll be able to make out that yes, if for example, if a well is after hybridization gives red color, so this means that this sample is expressed more in this cell type. And if a well gives a blue color or a green color, so that means that the control sample is more in this well. So that means that the transcript level or the mRNA that we are looking for is more in this well. And if we combine both the wells and both the wells expressed equal amount of both the DNAs, so what happens is the mixed color that was made after the combination of these two colors will be formed, and this will, I think, that will yield in a yellow color. So that will give us the idea that the RNA that is being expressed in both the samples Then we'll move forward to the next question. Hmm. So the chip is made up of either glass or silica. Hmm. So this is the next question. So a plant was grown under four different types of stress. So that can be anything. Stress from 1 to 4, and RNA was isolated from the trichome of the plants. So trichome is basically the epidermal shoot hair that is present on the plant, and RNA was isolated from that, and microarray analysis was done on each sample for different genes. Result is shown in the figure. So as you can see that this is the mRNA content for after microarray analysis. So this is the result that we got after analyzing the data that we got from microarray. So this is for sample 1, that is in stress 1, and likewise stress 2 sample 2, stress 3 sample 3, and stress 4 sample 4. So blue color represent now blue color represents the low expression of the genes, and red color represents very high expression of the genes, and the intermediate colors that follows, that is the black, that dark red, and all these are intermediate expression of the genes. So, and here are the name of the genes that are written here on the right side. So you have to determine what uh, hello, am I audible? Okay. So you have to determine which gene is expressed more in all the types of stress conditions. Okay. Any more answers? Okay. So gene I is the answer, and it is expressed more in all the types of stress because it shows that all the four lanes, that is in stress 1 to 4, it shows very high expression as compared to the other genes. So it is showing very high expression. Okay. So any doubts in this question? Okay. Just a second. Yeah. So now let's move on to the next question. So the question is, which of the following is an incorrect statement? So is my question visible? So the options are: SAGE and DNA microarrays are both high throughput techniques that determine global mRNA expression levels. Second option is: studies have indicated that the gene expression measurements from these methods are highly inconsistent. The third option is: SAGE does not require prior knowledge of the transcript sequence. And fourth option is: DNA microarray experiments can only detect the gene spotted on the microarray. So, okay, any more answers? So, moving forward, the correct answer is option B. Hmm. So, basically, first we'll understand what is SAGE. So, in case of SAGE, what we do is we basically isolate the mRNA and reverse transcribe into a cDNA, followed by we generally break them into we just have to add the tags on that cDNA. So we add those tags, and those tags basically anchor to the beads. And after that what we do is we generally put a tagging enzyme that basically cleave that tags that cDNA added on the bead, and then cleaves that. So after that we take tags from two beads, and then we generally ligate them, and followed by what we do is we then religate them, and that forms a die tag. And using a die tag we amplify using different primers that is on the for the adapters. And after that we usually form a concatemer, and we clone into a vector and then sequence it. So that basically gives us a this a relay kind of system. Like this is gene 1 RNA, this is gene 2 RNA, this is gene 1 RNA, and like the number of RNAs that are joined in this sequence are identified using different primers and different probes for different genes. So for from that we find out that yes, this gene is overexpressed in that mutant or like that. So this is how SAGE works. But what happens is that in case of this technique, different types of RNAs are just a second. And that they basically what they do is they form a concatemer that is a sequence of RNAs joined together of different genes. So after that they sequence and they hybridize with the probes of specific genes, and they find out that and they find out that which gene is overexpressed in the cell or the cell under consideration. Okay, so but what happens is yeah. So what happens is there is a lot of mRNA loss from these methods, being it SAGE or the microarray, because of the fact that if we are analyzing from this method, there is very non-spontaneous ligation, as well as the sometimes the RNAs that are expressed very less are not considered in these cases. That's why we are saying that the analysis done from these experiments is highly inconsistent. Okay, so any doubts still here? Hello, am I audible? Okay, so let's move on to the next question. So, the reverse transcription polymerase chain reaction can be used to option A, visualize the spatial distribution of the transcript in a tissue section. So, option B, quantify protein content in a tissue. Option C, study the chromatin structure, or option D, detect the presence of transcript in a tissue. So, reverse transcription PCR is basically used to detect the presence of a transcript in a tissue. So option D is correct. So we'll study about what is reverse transcription PCR and how it's done. So basically what we do is we generally isolate the mRNA from a cell. So we first isolate the mRNA from the cell, and as we know, in all the eukaryotes there is post transcriptional processing. And what happens is, like, there is capping, and there is a poly A tail, and there is splicing, that is the removal of introns. So here we are using this tail thing to generate a cDNA from the mRNA that is formed. So the mRNA will have a poly A tail, like you can see in the image, that it has a poly A tail. And we can do one thing, that we can design some primers like complementary to this poly A tail, and you, like, this is a poly T, and this can be used as a primer to basically uh, form the complementary of this mRNA. Okay, and that is we what we call it as a cDNA. And so and this whole reaction is done by reverse transcriptase enzyme. So here the reverse transcription happens, and after that what is done is, after completing this, we get a RNA DNA hybrid, and using this the reverse transcriptase only have the ability to degrade this RNA, that is, it has a RNase activity. So it degrades the mRNA that is complementary to the cDNA that is formed. So after that we get a cDNA, single stranded cDNA. So after that, what we do is we generally amplify this cDNA using gene specific primers, and the Taq polymerase that we use generally for the normal PCR. So this is how RT-PCR is done, and from a mRNA sample we generally get a double-stranded DNA, and that too amplified. Okay, so this is the basic procedure and principle of RT-PCR. So any doubts here? Hmm. Yes. Hmm. Yeah, so reverse transcriptase has activity of, first is the RNA dependent DNA polymerase, and second is the RNase activity. So it has both RNase as well as the polymerase activity. So it helps in, yeah, actually it has this activity only when RNA is hybridized to a DNA. Otherwise it doesn't degrade the RNA. Hmm. Okay, any more doubts? Okay, if not, then we'll move forward to the next question. So the next question is, which of the following is an example of targeted suppression of... So yeah, the correct answer is RNAi. So gamma rays is basically used for the generation of mutations in the DNA, and RNAi is basically a form of mechanism that helps in suppression of the gene at the transcription and the post-transcriptional level. And tDNA insertion is not a method of suppression of the gene expression, and EMS is also used for generating mutations in the DNA. So option B is the correct answer. So now the brief mechanism of how RNAi functions. So like the normal process of transcription, like this is the DNA, and we form a single stranded mRNA. But like in other cells, or like we know that some genes are only expressed in some cells while the other cells do not express those genes, so they are being controlled with the process called as RNA interference. So what happens is, in those cases, a double stranded RNA is formed. So how is a double stranded RNA formed? Like this is the 5 prime to 3 prime end of the DNA, and the other strand is 3 prime to 5 prime. So if a RNA is transcribed from this first strand, that is the first RNA, and the second strand is the second RNA. So both of these RNAs will be complementary to each other. Okay, so after transcription, both of them can form a complementary interactions, and they form a double-stranded RNA, like it is mentioned in this one. So they form a double-stranded RNA, and following that double-stranded RNA, they use an enzyme called dicer. So dicer basically cleaves this double-stranded RNA into small RNA that is not more than 50 nucleotide long. And after that, what happens is a protein, argonaute protein, and which combines with the risk complex. So risk is RNA induced silencing complex. So they basically generate a small like towards 30 nucleotides, less than 30 nucleotides RNA, that is specific for a specific gene. Okay, so after that, what happens is they can either go to the epigenetic modifications of the gene, or they can just lead to RNA degradation, or they just go to the or they just bind to the other RNA complexes and inhibit the gene expression, and secondly they can lead to post translation inhibition of the protein synthesis also. Yeah, just a second. And it is a common mechanism in every organism, because like in plants, there is expression of some of the microRNAs that helps, or as well as that helps to inhibit some processes as well as activate some processes. So this is all about the RNAi mechanism, and if there are any doubts, I'm ready to help. Hmm. Yeah, so it basically is involved in defense, as well as it is a defense process in plants as well as in various other organisms to suppress the expression of genes that are cells because of this mechanism itself. Okay, any more doubts? So if not, we'll move to the other next question. So the next question is, during RNA in situ hybridization, proteinase K, that is a protease, is used to enhance cell permeability and remove nucleases which may otherwise degrade the transcripts. At what step proteinase K treatment is done during in situ hybridization? So the options are pre-hybridization, washing, antibody incubation, and detection. So, like, can you guys guess what can be the answer? Sorry. Yeah. Any more guesses? So yes, this treatment, proteinase K treatment, is basically used in the pre-hybridization step. So how is basically these things done? So we usually like take a tissue. So here it's a plant tissue. So first we take a tissue and divide it into small sections. So like this is the branch of the plant, or so we divide it in small sections, and then we usually like embed them into parafilm blocks. So what happens is, like, after that we generally have to fix the tissue. That is done with the help of ethanol and various other steps. So after that they are embedded in the parafilm. uh, this is basically gives the adhere to the tissue, so that we can get good sections out of that. So we cannot stain anything out of this whole mount or the whole part of the tissue. So we generally take sections out of it, and we generally place it into a cryotome or a microtome that helps in giving us very thin sections that can, from that we can visualize the proteins as well as the RNAs. So after that we generally sections these tissue that are embedded in the parafilm, and then after that we then do a pre-hybridization tissue processing. So in this we usually add proteinase K and other things that helps in the permeabilization of the cells, as well as degradation of various other like nucleases, proteases that does not degrade the or the analyte of the cell. So as well as detergents are also used to permeabilize the cells. So after that what happens is, the after permeabilization the RNA probe is added to the cell that have been permeabilized, and after that we generally do basic blocking steps so that the probe we have given does not bind to the unnecessary location, or it does not give unnecessary results. So after that we generally do the blocking as well as the incubation, and followed by mounting and analysis in the microscope. So this is how the basic hybridization technique is followed. So any doubt here? Hmm. Sorry. Hmm. So if not, then we'll move to next question. So which of the following statements is false about RNA in situ hybridization? So the first option is, it is used to detect nucleotide sequences in cells, tissue sections, and even whole tissue. So nucleotide it doesn't really detect. So the first option is correct. First option is wrong. Sorry. And then it is based on the complementary binding of a nucleotide probe to a specific target sequence of DNA or RNA. So nucleotide probe to a specific target sequence of DNA or RNA. Yes, this is also correct. because the the in situ hybridization is basically complementary binding of the probes or the nucleotide that we are going to find in the cell. Then the probes can be labeled with either radio, fluorescent or antigen labeled bases. Yes, this is also correct. These are different ways by which the probes can be labeled, and the ribo probes have identical sequence to the target RNA. So this option is false, because if we have to hybridize anything, like this is the nucleotide sequence and we have to find out this sequence in a cell, so we have to first like have the complementary sequence of this. That's how it will bind to this sequence. Okay, so the option D is false. And this is basically a representation of how this the final steps of the RNA in situ hybridization is done. So this is the mRNA that we have to find, and using this this is the probe that is labeled with digioxygenin. So this basically helps the antibody or the antibody that identifies this probe and helps us recognize that where this mRNA is present. So after that, a specific antibody is added that binds to this probe, specifically to digioxygenin, and after that, a color is substrate is added to the this antibody. This antibody is basically linked to an enzyme that converts a substrate to a chromogenic product that is identified by the color. So that increases the sensitivity of the assay. So this is how RNA in situ hybridization is done. So any doubts still here or the technique? Anyone? If not, then we'll move to the next question. So the question is, which of the following will be the best strategy to study the subcellular localization of a protein in planta? So we have to study the subcellular localization of a protein inside the plant. So we can use either transcriptional promoter reporter fusion, translational protein reporter fusion, Western blotting, or none of the above. Can you guys take a guess? Okay, any more answers? Yes, so the correct answer is option B, translation protein reporter fusion. And how this is done is basically, so there is a difference between the above two options, that is the transcriptional promoter reporter fusion as well as the translation protein reporter fusion. So what we do is, in case of a transcriptional gene fusion, we have a promoter that is common for both the genes. Okay, so there will be only one promoter that transcribes both the genes, but they have different ribosome binding sequence of their own. So the proteins they will form will be different and separate from each other. And this translational gene fusion is basically you remove the stop codon from the first gene and the ribosome binding site of the second gene. So you keep both the genes together, and that's how two genes will be fused. And here we use generally GFP as a reporter gene that is fused with the protein, and that helps us to identify the subcellular localization of the protein in the cell. So this is how different proteins have been synthesized and looked upon in various organisms. Okay, so any doubts in this question? So let's take an example of a bacteria. Okay, so Yeah. So what we do is we generally use this genetic engineering techniques in this case. So I'm talking about the translation protein fusion. Okay. So like we have to study a protein that embeds in the membrane. Okay. So what we'll do is we will make a vector that contains a promoter, and after that promoter there will be a gene of that uh, transmembrane protein, and this gene will be linked to a reporter gene like GFP. So when this vector will go inside the bacteria, so what happens is this will like express inside the bacteria because it has both promoter as well as the ribosome binding site. So this whole region will translate into bacteria, and after processing it into after whole processing as well as the localization, it will go into it will be embedded in the membrane. So after visualization under a microscope, it will be found that the reporter gene is near to the membrane or is embedded in the membrane. Okay, so this from this we can understand that yes, the protein that we expressed is a transmembrane protein. Okay, so this is how basically these experiments are done. Hmm. Yeah, but in case of a transcriptional fusion, actually what happens is when a protein like a gene is transcribed, so there is only a single transcript formed. Okay, but they have a different ribosome binding site from where they are translated. Okay, so for example, here in this case only, if we see the first gene that is being translated has a stop codon here, and after that there is a ribosome binding site of the second gene. So after they are translated, both the genes will be separate. Okay, so there is no link between them. That's why they cannot be used for subcellular localization. Hmm. 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 So in both the cases, there will be movement of that gene or the protein. But what happens is in the transcriptional fusion, there will be no fusion of the protein. There is only the RNA that is formed is fused for both the genes. Yeah. Yeah. Yes. Hmm. Hmm. Okay. So any more doubts? Now, what your doubt is, it will be cleared by the next question. So, the next question says you are analyzing the movement of a transcription factor in different regions of the roots. Okay, to find out whether transcription factor move between two cells at the transcriptional or the post-transcriptional level. So, we are finding that whether the transcription factor RNA moves to different cell or the transcription factor protein moves to different cell. Okay, so we use both transcriptional promoter reporter fusion as well as translational promoter reporter fusion techniques using GFP. So we are using the same constructs like I have shown you before. So, but you find that the transcript. Hmm. So the transcription factor RNA is only limited to some cells, while the protein is moving to the other different cells. So the inference on the basis of above observation. So the basically inference will be that RNA does not move, but the protein moves, and that can be visualized using this image. So, like if we have the GFP coding region in the transcriptional fusion, it will be localized in this area only. Okay, but if we have fused the transcription factor with the GFP, so it is a translational thing, a translational fusion. So we can see that the transcription factor is moved to different cells, and we can visualize this because the transcription factor is fused with that gene. Okay. Hmm. Okay, so let's move to the other question. Yeah. Yeah. Okay, so what we can do is actually we generally insert anything on the stage of gene. Okay, we doesn't do anything on the stage of mRNA. We study that stage at mRNA. Okay, so we generally if there is a plant that is of stage A of their development, and in initially we have introduced a gene that is fused with a GFP reporter. Okay, and this gene only expresses when it's in developmental stage A. So when the plant grows, this protein will form along with the GFP, and it will show some signal. Okay, so that's how we can study this protein, that it's been expressed in that particular stage. Yeah, that's no, actively dividing cells. So we can do that also. But if we have to check a particular stage of the plant, so yes, we can use that also. Hmm. Okay. So any more doubts? So if not, we'll move to the next question. So CRISPR is, it's a gene editing tool. It's a family of DNA sequence derived from viruses in the genome of a bacterial host, an endonuclease, or a disease. So, any answers? Sure. Any more answers? Actually, it's not a gene editing tool. Okay. So, it's basically a family of DNA sequence that is derived from viruses in the genome of bacterial hosts. So, CRISPR basically stands for clustered, regularly interspersed palindromic sequences. Okay. Palindromic repeats. So, along with Cas9, so CRISPR Cas9 is a gene editing tool, but it's only a sequence that recognizes a particular sequence inside the host, and this has been derived from viruses inside the genome of the bacterial host. So, bacteria basically use this CRISPR as a immune response towards the viruses. Okay. Yeah, so And if there is only Cas9, so it will be an endonuclease. So Cas9 is an endonuclease that recognizes these sequences and cleaves at that point. Okay, so this will be better understood with this image. Just a second. Yeah, can you all see the image? So this is the genomic DNA. Okay, this is the guide RNA that we use for guiding the Cas9 to a specific region. So this region is specific to the DNA to be cleaved. Okay, and this is the spacer arm, and the Cas9, it's an endonuclease. It recognizes these PAM sequences. These are these are basically protospacer adjacent motifs. Okay, so these Cas9 recognizes PAM motifs and cleaves the DNA at this region, and it is a double-stranded break. Okay, so this is how a CRISPR-Cas9 system helps, and we use this sgRNA or the guide RNA to cleave different sequences inside the genome of any organism. So it is being used widely to generate many recombinant or recombinant GMO. Okay, so any doubt till here? Anybody else? If not, we'll move to the next question. So, yeast-2 hybrid assay is used to study DNA-DNA interactions, protein-protein interactions, DNA-protein interactions, or RNA-protein interactions. Okay, any more guesses? So yes, the answer is protein-protein interactions. If the question would have been yeast-1 hybrid assay, then it would be DNA-protein interactions. Okay, so what is usually done in this case? So we have to study the interaction of two proteins. So how can we study that? We usually do this using a bait and a prey protein. So like we all know, in case of a transcription factor, okay, so transcription factor basically helps in the transcription of the DNA. So this is the DNA, and so this is the DNA, and transcription factor binding site is here. So transcription factor has two components in it. So one will be the DNA binding domain, and another will be the activation domain that activates the other transcriptional machinery further. So what happens is we fuse this DNA binding domain to the one interacting partner of the protein, and the other activation domain with the other interacting partner protein. So, and we usually do it in two different plasmids. So one plasmid will have the DNA binding domain with the protein X, and the other plasmid have the activation domain with the protein Y. So this is how it's translated in the organism. Okay, so if this both X and Y interact, so what will happen? So, and then we use a reporter gene like GFP or beta galactosidase to prove that the two proteins are interacting or not. So this is the DNA. Yeah. So this is the DNA binding region of the cell. Sorry. Yeah. So the DNA binding region of the transcription factor will bind to the DNA at its site, and if both the proteins are interacting with each other, then the activation domain can activate the transcriptional machinery, followed by the expression of the reporter gene. This will give us a result such that we can show that both the proteins are interacting in the cell or not. Okay, so this is how we use this yeast to hybrid assay in case of protein protein interactions. So any doubt till here? Hmm. Sorry, repeat once. Hmm. Sorry, I could not get your question. Hmm. Hmm. Oh, so actually what we are doing is, here we have to study whether these two proteins X and Y are interacting after they are translated. Okay, so this we basically do a translational fusion. Okay, such that we have one, that is the X protein is bind to the DNA binding domain, and the Y protein is bind to the activation domain, and if these two interact, then only the reporter gene will be expressed. Hmm. Okay, so we are not doing anything at the mRNA level here, we are doing at the protein level here to find out the interaction of the two proteins. So any more doubts in this? Yeah. So let's move forward to the next question. Yeah. So this is basically the analysis of the yeast to hybrid assay. So a scientist is studying effect on interaction of protein A with the other protein B, which is mutated. So he developed two mutants of the protein B, that is mutant 1, mut1, in which arginine 213 is replaced with histidine 213, and mut2, in which aspartate 450 is mutated with glutamine 450. Okay, so here the scientist is basically checking the interaction of protein A with mutant protein B, and he has generated two mutants. One is the arginine mutant and another is the aspartate mutant. Okay, So he used a yeast to hybrid assay using beta mute 1 interaction with protein A. The beta galactosidase activity was more as compared to the wild type protein. Wild type protein is the one that does not have any mutations in the protein. Okay, so protein B has the normal sequence and have no mutations. Okay, and this activity is of mute 1 is more in case of mute 1, and the protein A is more as compared to the wild type as well as the mute 2. So what can you infer from the above statement? So the options are: interaction between protein A and mute 1 is strong. So interaction between protein A and mute 2 is strong, and interaction between both the proteins is and the wild type is strong, or none of the above. So can anyone guess or analyze what the question is? So anyone? So what the scientist did was, he has a protein B that he is checking the mutations. Okay, so we have three variants of B here. One is the wild type, one is mute 1 that have arginine mutated to histidine, and another is mute 2 that have aspartate mutated to glutamine. Okay, so what is what the scientist did was, he did an yeast to hybrid assay for to check the interaction of these three protein B with protein A. So now he put protein A with this, and to check the interaction between these two, he used a reporter gene, that is beta galactosidase. And he found that in case of mute 2, there is more activity as compared to the wild type as well as the mute 2. Okay, so what can you infer from this information? So as yeah, anyone wants to answer? Yes, so there is strong interaction between the protein A as well as the mutant one. That's why it's showing high beta galactosidase activity. Okay, so this can also be inferred that the mutation that the scientist is studying helps in interaction or improves the interaction of protein A with protein B. Okay, as compared to the wild type. So the answer is option A. Okay, so has any doubt in this question, or do I need to explain this again? Mm, sorry, can you? Oh, so we can use any other things, but in case of yeast to hybrid assay, we have a reporter gene. Okay, and that too beta galactosidase. We do not use any reporter gene in other interaction assays. Hmm. So anyone who have not understood this question? Okay, so let's move on to the next question. Yeah, so you use a technique to study the interaction of a protein with a transmembrane receptor. So you use a reporter gene of which half part is attached to the protein and other half is attached to the receptor. Okay, so here we are using a reporter gene, and of that gene half part is attached to a protein A and half part is attached to a protein B. Okay, so if both these proteins interact, then you get a functional reporter assay after that. So these two proteins have been in conformation after these two proteins interact. So what is this technique called? Is it yeast to hybrid system, co-immunoprecipitation, biomolecular fluorescence complementation, or Western blotting? So anyone take a guess? Okay. Anyone else? So this is not yeast to hybrid system. It's biomolecular fluorescence complementation. Okay. So what happens in this case is we usually have a protein P1 and this is attached to the half part of the reporter gene. So here we are representing it as YFP, that is yellow fluorescent protein, and that is the N terminal of that protein. So the portion towards the N terminal of this YFP is attached to protein P1 and the half part of the protein YFP is attached to the protein P2, the other interacting partner. So if these two proteins interact, okay, so this YFP will get a stable conformation and it will give a signal at 520 nanometer laser, that its excitation wavelength. Okay, so this is how if the proteins interact, the YFP forms a stable conformation and it's active now. So if we put a laser of 520 nanometer, it will give a signal. Okay, this is how we use biomolecular fluorescence complementation. Now, how it's different from yeast to hybrid system is that in case of yeast to hybrid system, we use two different proteins that are basically fused with the DNA binding domain and the activation domain of the of a transcription factor that we generally use for the that is used in yeast. Okay, but here the reporter gene is itself like distributed among those. So directly the signal is visible after the proteins interact. But in case of yeast to hybrid system, if the proteins interact, then the reporter gene is synthesized. Okay, so this is the basic difference between yeast to hybrid system and the biomolecular fluorescence complementation. Sorry. So any doubts still here? Okay, any doubts anyone? So we are moving to the next question. Yes. So in order to discover specific cell type marker, the scientists generated a collection of transgenic lines with random insertion of a promoter-less reporter gene. Okay. So here there is a promoter-less reporter gene that we are using and screened for specific reporter activity in the domain of interest. So this technique is called as, these are the options. Any more answers? So yes, this technique is called as promoter trapping, and how this is done is that there are different trapping techniques, like there is enhancer trapping, there is report promoter trapping, and there is gene trapping. So this is the normal genomic DNA that we are studying. This is the data box, that is the promoter, and this is the enhancer that enhances the transcription of the like using this data promoter. Okay, now what we do is an enhancer trap. We use a promoter with the reporter gene separately, and using this inbuilt enhancer, this reporter gene is also synthesized. Okay, so this is the enhancer trap. In case of a promoter trap, we use a reporter gene that is embedded in the gene itself that was there initially, and it does not have any promoter. So when this enhancer is like this promoter synthesizes the transcript, the whole gene synthesized, and the reporter is also synthesized along with this. Okay, and in the gene trap mechanism, what we do is we generally use the splicing mechanism, and the splice acceptor site is introduced into a reporter, and that is like during splicing, this reporter gene is introduced into the normal gene of the plant. So after that, what we get is a gene that is being fused with the reporter after the splicing has been done. Okay, so this is the gene trap mechanism that is takes place. So this is how different trap mechanisms is used inside the plant. So any doubt still here? Yeah. Hmm. Mm. Sorry, can you repeat? Yeah, we can use agrobacterium mediated insertion also, because that's a random process. So that's how we generate transgenic lines with random insertions. Hmm. So like there is some time point that after infection of the agrobacterium, the we can check whether the tDNA is inserted into the plant or not. So I have to check on that time point, but yes, there is a time point that after that we can check, and using different tDNA insertion platforms, we can check using PCR, using the like we discussed in the last lecture. So we can check the tDNA insertion into the plant. Uh, when can we apply any method or like that? You're asking. Hmm. Okay, so actually, it takes around after infection, 3 days or 1 week and after that. So we generally use that time point. And because this all things agrobacterium thing is we are doing in a culture, so we generally don't like put them on a large time gap. So we this is very small time gap, like in a day or two we isolate different cells from that culture, and we generally look for the tDNA insertion into that. So this is how it's done. Sorry, can you repeat? Okay, so what we can do in that case is, like we have discussed in the last session, that we can use a specific primer for tDNA. Okay, so suppose this is the tDNA insertion site, and we have a specific primer for this tDNA that is being inserted. Okay, so after we isolate the DNA and do the PCR using this, and the reverse primer that we are using is outside the gene, and use like various primers can be used for the reverse primer, because we do not know where the tDNA is inserted. Okay, so after this we put on an agarose gel, and we find that this particular uh, region that we are observing is based on the tDNA insertion only. That's how we can look for tDNA insertion inside the cell. Hmm. Hmm. Okay. So, any more doubts? If not, we are moving to the next question. So, yes. So, a scientist analyzed the expression of a gene involved in development of plants in a plant in roots, leaves, and flowers. So, he is using QRT-PCR technique, and he got the following graph. So, the orange line represents the root, the expression of the gene in root. The blue line represents the expression of the gene in leaves, and the purple line represents the expression of the gene in flower. So, looking at this graph only, can you tell whether which organ has the least expression of that gene? Anyone? Okay. Any other answers? So actually what happens in qPCR, so this is the final graph that we get after amplification. So like it gives the values on the basis of a CQ. Okay, so less CQ means more amount of DNA or RNA present in that sample. ठीक है? So what happens is, in this graph, the graph that is basically starting earlier has less CQ, followed by this and followed by this. So the least expression will be there with the high CQ sample. Okay, so this orange line shows less CQ, followed by this blue, followed by this purple. So this means that the purple line has less expression of that gene. That's why its CQ is more. So here. So the gene that have least expression is in flowers. Okay, so this is the option. So CQ is basically like the it basically gives the number of cycles after which the gene is started to amplify. Okay, so this can be understood with this figure. So like we have the RNA isolated and we generated a cDNA out of that using reverse transcription. Okay, so now we are doing the real-time PCR. So what happens in this Q in QRT means real time. Okay, so RT means reverse transcription, but Q means real time PCR. So we just check the number or the value that this DNA or this RNA is more in that sample or less in that sample. So how do we do that? We use generally two basic methods. First is the cyber green and the other is the Tachman probe. So the cyber green is the dye that basically binds to the double stranded DNA. Okay, so when this DNA starts to amplify, so this cyber green binds and give the signals. So if the DNA that is present more in the sample, then the levels of this cyber green or the signal of this cyber green will start at the low cycle range, hence the low CQ. Okay, so if there is more DNA like less DNA sample, less DNA amount in that sample, hence this cyber green will give signal after a specific number of cycles only, hence very high CQ. Sorry. So hence in that case the CQ will be high. Okay, and the other thing is the Tachman probe. What we have in this case is a probe. So this probe is like in a gene, this probe binds specifically, and it has two parts. One is the quencher and one is the fluorescently labeled thing. So the quencher basically quenches the fluorescence of this part. So if they are near to each other, the fluorescent tag cannot express itself or cannot excite itself. And when the DNA polymerase comes during the PCR amplification, so what happens is uh, the probe is cleaved from between and the quencher is separated from the fluorescence tag, such that the tag can now be excited and then it gives the signal. So if the like the same thing happens as in cyber part. So if the the number of DNA is low, high, so this will give signal very early, hence very low CQ. So the amplification started at a lower cycle of the PCR. Okay, so this is how the Tachman probe works, and this is repeated to about 30 cycles, and after that we get signals in the form of this graph or the CQ. Okay, so this is how the QRT-PCR works. Now, understood everyone, or any questions? If anyone has not understood, I can repeat the process also. Hmm. Sorry, can you repeat? Okay, so cyber green is basically a fluorescent dye that binds to the double stranded DNA. Okay, so when the DNA is single stranded, it will not give any signal. But when the DNA is double stranded, it will like bind and adjust itself into the duplex that is formed. Okay, so double stranded DNA present in the sample. For example, like we have about 14 copies. I'm just taking the example. The copies are very much higher as compared to this number. So one sample has 14 copies of the DNA at the time of initiation. Okay, so when this process of PCR starts, so it starts amplifying at the cycle of uh, like fifth cycle, it starts amplifying. Hence this will give a CQ value of that is very less. Okay, now the other sample that has DNA copies of only 8. Okay, so this will start giving the signal at around 12 or 13 cycle. So this CQ will be higher. Okay, so this is how this cyber green help, like all the real-time PCR probes helps in determining the number of cycles at which the DNA is starting to amplify, and that depends on the copies of DNA that is present in the sample. Okay. So any more doubts? If not, then we'll move to the next question. So how can we check DNA protein interactions in a spatiotemporal way inside the cell? So here are the options. Can anyone take a guess? Sorry, can you repeat? Sorry, I could not understand. FRET, option A. Okay, anyone else? Yes, so the answer is FRET, that is fluorescence resonance energy transfer. Okay, so how this basically takes place is that we have a DNA molecule that is tagged to a fluorescent tag. Okay, and it expresses at it excites at a wavelength lambda 1, suppose. Okay, so now if there is a protein that binds to this DNA and it sits here, and it is also tagged with a dye that expresses that excites at Y. Okay, so if this protein interacts with this DNA molecule, then this like considering this to be the donor and this to be the receptor, so if we excite the protein this excites at lambda 1, so we give lambda 1 to this protein, and we check for the emission wavelength of this protein. Okay, so if this in protein and DNA are interacting, then the emission from this particular reporter will be there. Okay, and if these two are not interacting, then there will be no emission from this protein, and only emission will be observed from this protein only. Okay, so this is how we call it as a FRET on state, and if they do not interact, then it is a FRET off state. Okay, do I repeat this portion, or have everyone understood? Sorry, can you repeat? There is a lot of background. Mm, Sorry, I didn't understood the last line. Hmm. 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 Okay. Hmm. Okay. So here we have to check whether, like for example, this protein, like this red one, is a protein that is a transcription factor. Okay, and it is binding to the DNA. Okay, we are not analyzing anything that it is expressing or repressing anything. We are just showing the interaction that this protein is interacting with DNA or not. Yeah, just binding. Hmm. So, like few other considerations that are to be kept in mind to study FRET is that the distance between the donor and the receptor is not more than 100 Armstrong. Okay, it must be less than 100 Armstrong, such that these two will be near to each other, and then only the excitation of this protein can lead to energy transfer to this reporter. And the second thing is, if we check on the graph of these two proteins, so this is the emission excitation of the protein A, that is this one. So it excites at lambda 1 and emits at lambda 2. And the protein B, it excites at lambda 3 and it emits at lambda 4. So the emission wavelength of protein A must coincide or overlap with the excitation wavelength of protein B. Then only this FRET can happen. Okay, so this is how we study the FRET. Yeah. So understood everyone? Okay. Now we move on to the next question. So, the technique used for understanding transcriptional regulation of genetic expression in vivo. So, which technique from these can be used to understand the transcriptional regulation in vivo? Can anyone make a guess? Sorry? Okay, any more guesses? So, yet it's chip assay, that is chromatin immunoprecipitation. Okay, so this is, sorry, chip will be the answer, not FRET. Okay, so this will be option C. No, actually, we have to first fix those things in case of FRET. So that can be in vitro as well as in vivo both. So that is not in vivo thing only. We generally don't use FRET in vivo. We generally use FRET in vitro only. So that's why uh, it's not the correct answer. Okay. So what we do in this case is, the cells are generally what we do is, the cells are grown and the DNA that we have to check is first bound with the protein that we have to check, and after that we use a specific antibody towards the protein that is bound to the DNA. Okay, so and then this protein bound to the antibody is basically precipitated, and after that the protein antibody complexes are moved on beads. We check for the DNA samples, and we sequence them and followed by their analysis. So that's how we find that the transcription factor or the protein is binding to the DNA that we are looking for. Okay, so this is basically the chip assay that we do. So any doubts still here? Okay, so let's move on to the next question. So you are using a DNA's footprinting assay to detect the interaction of two transcription factors to the genes involved in root of plant under salt stress. So we are looking for a gene that is involved in the roots of plant that are in the salt stress, and we find that two transcription factors can bind to both this gene under the salt stress. So we have to find which transcription factor bind to this gene using DNA's footprinting assay. So this is the gel image given, and these are the sequences that are given. So one is the ladder, second is the transcription factor 1 bind to the DNA, third is the transcription factor 2 binding to the DNA, and fourth is the control. Okay, so what can you infer from this graph? So can anyone take a guess? What can be the answer? Sorry, could not understand what you are saying. Okay. Any other else? Anyone? So what we do in DNA's footprinting assay is that we take a DNA and we generally add the transcription factor that or the DNA that we have to study, that this binds to this part of the DNA. Okay, then we apply DNA's treatment and the region is chopped off other than the region that is bound to the DNA. Okay, that is the protein that interacts with the DNA. Leaving that region, other regions are chopped off. So this is how you get these much bands in this well. That this is the ladder. Okay, and this is the control, that is only the DNA that is being used under the experiment. Okay, now in case of lane 2 and lane 3, we add lane 2 have TF1 and lane 3 have TF2. Okay, so here we check the interaction of TF1 with TF2, and what we find that if any of the TF1 or TF2 will be bind to this DNA, okay, so this region that is empty here, this means that a protein is bound in this region on the DNA. Okay, and this is not visible in this part, and there is no empty region in this lane. So this signifies that the TF1 is binding to the DNA that we are studying. Okay, and that can be involved in the roots of under plant stress. So the answer is A, that TF1 is binding to the DNA. That is why it's showing this space in this gel assay, and this is how we are studying that TF1 is binding to the DNA. Sorry. Hmm. 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 So here what we are doing is, on the DNA, if the transcription factor binds, so after DNA's treatment, the region that is left over, that is not bound to the protein, will be chopped off. So only this region will not show any band in this part. Okay. Hmm. So any more doubts regarding this, or shall I explain the process again? If not, then we'll move to next question. So this is colony hybridization technique is used for transformation of a clone, for checking protein-protein interactions, for checking protein expression levels, or for clone conformation. So can anybody take a guess? So this technique is basically used for clone conformation. Okay. So how do we do this? So for example, like we transform bacteria or some plant cells with a particular DNA or a vector that contains the gene of interest. Okay, so here are these transformants on a plate. So these are the cells that have that DNA. Okay, so now we have to check whether we have the gene of interest into them or not, or it's the empty plasmid. Okay, so now we move on to the other plate or a membrane, and then what we do is we lyse and wash them, as well as fix them, and then we add a probe that will hybridize to the specific region, and then we do a analysis, whether it is autoradiography or fluorescence. Okay, so then we check whether the gene of interest is there in the cell or not, and after that we select clones from here, and we further analyze them or use them for further experiments. Okay, so this is the basic mechanism of colony hybridization that we use. Okay, so any doubts in this question? If not, then we'll move to the next question. So a researcher have to check the expression levels of a microRNA in a specific developmental stage of a plant. So which technique can we use to analyze that? Okay, so here are the techniques given: northern blotting, southern blotting, QRT, PCR, and or both A and C. So which technique can the researcher use? Can anyone take a guess? Okay, anybody else? So yes, both A and C are right, and we have studied about QRT PCR in the slides before. So here we will look briefly upon the northern blotting. So how it's done is, we generally isolate RNA. So it's a cell that we are considering, and we isolate RNA from this, and we generally run into a gel. So it's a denaturing agarose gel. Why denaturing? because RNA generally forms secondary structures with itself. So we use a denaturing condition so that it cannot form hydrogen bond with itself and cannot form a secondary structure that will hamper our further. What we do is we run this on a agarose gel, denaturing agarose gel, and then this whole gel is transferred onto a membrane. Okay, so now this membrane is a RNA binding membrane, that is nylon or nitrocellulose. Then uh, to transfer this, we generally keep it in a solution that through capillary action transfers it to the membrane. And we use blotting paper, and there is a weight on above that so that it will get transferred onto the membrane. After that, when the bands are transferred onto the membrane, we generally use a probe, that is a nucleic acid probe, for detection of a particular mRNA that we have. Okay, and then we expose this to either autoradiography or fluorescence, as like the case may be, and then we find that this particular RNA is expressed more in case of particular gene, a particular case, and this is expressed in case of this is expressed less in case of other cases. Okay, so this is how we use a northern blot technique. So any doubts still here? Okay, so this was the last question for the session, and if there are any doubts in the questions that we have discussed today, we can discuss it. Or if anyone has doubt in the this week's session. Hmm. Okay. Hmm. Okay. So what uh, what is the specific doubt in that? Hmm. Just a second. Just a second. Just a second.